Biology 1108, Presentation Number 7, Protists. So we continue on our trip through biological diversity. We've taken a look at the bacteria. We've taken a look at viruses that technically aren't living, but still are part of the big picture. Let's talk about the kingdom Protista. I will remind you, it wasn't too long ago that we mentioned that kingdom Protista is considered to be a polyphyletic group. It's problematic. There's an awful lot of different types of organisms in kingdom Protista, and they share basic characteristics. They're all eukaryotic. They are, for the most part, single-celled organisms, although there are some exceptions. That's why the asterisk is there. They are, for the most part, motile, although there are some exceptions. That's why the asterisk is there. And there's a hodgepodge of some autotrophic organisms and some heterotrophic organisms in the kingdom Protista. Historically, it was a fairly easy group to come up with, but as scientists have looked at the kingdom Protista, they realize it's really a jumble of a lot of different unrelated groups of organisms. And hence, we're going to say it's polyphyletic. If you take a look at the illustration at right, you will see that the protists are highlighted in yellow, and I see there are several different possible origins here. So technically it is a polyphyletic group, not a monophyletic group. Now, what we're going to be doing is we'll be taking a look at how we actually resolve the kingdom Protista evolutionarily, and we are going to depart from what amounts to being standard evolutionary procedure. So, Dr. Knaus, what do you mean, depart from standard evolutionary procedure? Well, here's the deal. Evolutionary procedure generally involves resolving a group into evolutionarily cohesive taxa, or groups. When biologists have taken a look at the kingdom Protista, they can resolve 5 groups within the kingdom Protista that represent evolutionarily cohesive groups. Those 5 supergroups are listed here: the excavata, the chromalveolata, the rhizaria, the archaeplastida, and the uniconta. Now, don't worry, I am not going to ask you to memorize these names. I'm not going to talk any more about them beyond what I see right here. And the reason is because this is one of these cases where evolutionary biology kind of gets in the way of practical value. See, the practical value for any scientist is to be able to just very quickly be able to glance at an individual organism and know exactly where it fits in the overall scheme of things. I can take a look at any protist under the microscope and I can tell you exactly where it fits in the grand scheme of things. I can tell you whether it's a member of the phylum Flagellata. I can tell you whether it's a member of the phylum Sarcodina. I can tell you if it's a ciliate. Now, the thing is those groups I've just mentioned are the ones that you and I are going to be learning. They don't necessarily fit into the evolutionary scheme of things. In this case, the 5 supergroups are actually evolutionarily consistent, but the thing is there are very limited practical value. So I see here, my last bullet point tells me that these 5 supergroups are largely based on evidence of endosymbiosis. Now, that's nice, and it's really important that I understand, yes, the kingdom Protista consists of at least a number of evolutionarily cohesive groups, but from a practical standpoint, I need something a lot more easy to be able to glance at any given one protist and know exactly what group it belongs to. And I'm not worried about the excavata, the chromalveolata, the rhizaria, the archaeplastida, or the uniconta, because those aren't terribly practical. So let's talk about the traditional means of classifying the kingdom Protista. All right, this practical or traditional classification of the kingdom Protista has been carried out for literally 100 years. scientists have been looking at protists under the microscope, and they've been grouping them into these very, very practical groups, which are of maximum efficiency and maximum ease. And that's really the benefit in traditional classification. See, right now, I guarantee you that 99% of biologists still use the traditional classification of the kingdom Protistae, even though it defies the evolutionary classification I just mentioned. So, when we take a look at the kingdom Protista, they fall into three general groups. The autotrophic protists, that is, the photosynthetic protists, are commonly referred to as the algae. The heterotrophic protists, and these are protists that are not capable of making their own food molecules. Sometimes they're called the colorless protists, are collectively referred to as the protozoans. And then finally, there is a group of protists that resemble fungi, and they are called the fungal protists. And they are heterotrophic as well. And we will take a look at each of these groups in various levels of detail. Our first group to look at are the algae. The algae are classified into a number of different divisions. These are groups, and they're largely based on photosynthetic pigments, what the organisms look like, and details of cellular structure. So at the very bottom of the slide, classification based on photosynthetic pigments, morphology, and ultrastructure, which means cellular structure. And these names, I want you to practice name recognition. I'm never, ever, ever going to ask you to come up with these and do a fill in the blank on them, but I need you to know that there are these divisions that exist. The Rhodophyta, rhodo is a prefix meaning red, so these are the red algae. They have photosynthetic pigments that make them reddish in color. The division Chlorophyta, which are the green algae, which incidentally are ancestral to our land plants. The division Phaeophyta, which are the brown algae. And perhaps the most important thing is to remember that these three groups of organisms, the red algae, the green algae, and the brown algae, are generally referred to as seaweeds. These are multicellular protists. Now, the remaining groups I see on this slide are technically single-celled organisms, and they are commonly found in a variety of marine and freshwater environments. There are the Chrysophyta, or the diatoms, the Purophyta, the dinoflagellates, and the Euglenophita, the euglenoids. Once again, I'm going to ask you to practice name recognition. Most of you guys will never ever encounter these ever again, unless you wind up going into some form of environmental science where you're looking at samples of lake water, pond water, or stream water, and so you're going to need to be able to be familiar with these names. But at this point, please make sure that you know that traditional classification of Kingdom Protista, as far as the algae is concerned, is based on photosynthetic pigments, the color of the organism, morphology, what the organism looks like, and ultrastructure, that is, internal structure of the individual cells. Now, most of our focus on classification will be on the protozoans. There will be a number of slides that will focus specifically on these different phyla. So this is kind of a bird's eye view of where we're headed with the protozoans. You will remember that the protozoans are the heterotrophic members of the kingdom Protista. They fall into these four different groups. The phylum Sarcodina, which are the amoeboids, the phylum Mastigophora, which are the flagellates, and the phylum Ciliophora, which are the ciliates, and then finally the phylum Apicomplexa, which are commonly called the sporozoans. We will be touching on these in individual slides. This is nothing more than, like I said, a bird's eye view of where we're headed. And I certainly would like you to know that these protozoans are classified primarily on the basis of their motility, their morphology, and their life cycles, which is a lot more easy to handle than some of the detailed stuff that are associated with that supergroup classification that we saw previously. okay, for the sake of being complete, we'll include our fungal protists here. This is our third group. These are protists that resemble fungi. Now, they're generally classified into 3 different divisions. I just want you to practice name recognition here. I will probably never, ever ask you about any of these groups. You might, however, encounter them at some point. So the Oomycota, the Chytridiomycota, and the Myxomycota. Now, each of these cases, you will see the word mycota in the name, or in the common names, Oomycetes, Chytridiomycetes, and Myxomycetes, you will see the word mycetes. Important clue for you guys, even for those of you who are going into the medical field, if you ever see the word mycota or the word mycetes, the implication is you're talking about something that is a fungus or is fungus-like. Mycota or mycetes, words that imply fungus. But technically, the fungal protists are classified based on their morphology and their life cycles. And your job at this point is to realize that they are the third general group of organisms found in traditional classification of the kingdom Protista, alongside of the algae and alongside of the protozoans. Okay, so here's where we focus on the specifics of the protozoans. And you'll remember that I mentioned that the protozoans are going to be largely classified on their motility structures. So we're going to start with the phylum Sarcodyna. Sarcodyna are the amoeboids. These are single-celled organisms that have no specific definite shape, largely because they move through the agency of cellular extensions that are called pseudopodia. Now, the word pseudopod means false foot. So what I see is a picture of an amoeba, that is an organism called chaos, immediately below the bullet points here, and you see it's got these finger-like projections. And what will happen is there will be cytoplasmic streaming that will see the flow of cytoplasm into those projections, and that's, number one, how the organism moves, but also, number two, how the organism actually is able to eat. It will engage in phagocytosis by surrounding some particular food item of interest and internalizing it in the form of a food vacuole. Now, our examples of members of the phylum Sarcodyna include a free-living amoeba, chaos, specifically below, and then there is a pathogen called Entamoeba histolytica. A pathogen is an organism that causes disease. Entamoeba histolytica is generally transmitted through fecal material and will find its way into, for example, vegetables imported from other places that come into American supermarkets. And if you buy lettuce, or if you buy celery, or if you buy any of these vegetables that might come from some other place besides the United States, one of the reasons why you wash vegetables. You wash vegetables to get rid of this stuff that could be on the vegetable material, and you eat it, that Entamoeba histolytica gets inside of you. And it causes this nasty, nasty disease called amoebic dysentery, which is not just miserable, but it's incredibly difficult to treat and get rid of. The flagellates go by a number of different names. Probably the most traditional name is the phylum Zoomastigophora, and I know that's a mouthful, but these are flagellated cells, cells that are motile through the agency of flagella. They're also sometimes referred to as the phylum Mastigophora, or sometimes commonly known as the phylum Flagellata, Flagellata, and I've seen all three terms in use. But like I said, these are single-celled organisms that have flagella. Flagella, you remember, are long beating hairs. Now, many of these organisms are free-living. We are going to focus on some that actually cause disease. The two that I want you to be aware of is Giardia, which is found here in the United States, and trypanosoma, which is a pretty significant issue in developing countries, particularly on the continent of Africa. Giardia causes a waterborne illness called giardiasis, and it's generally as a consequence of fecal contamination. I see this hiker taking this long, cool drink from this gorgeous flowing mountain stream. However, there's probably a piece of bare intestinal sculpture up the hill that would contain spores of this particular organism, and it winds up coming down the hill, and the hiker drinks it and goes home and has violent diarrhea for 3 or 4 weeks and spends another 3 or 4 weeks trying to get rid of it. Giardiasis is a very nasty condition. And so the fact of the matter is if you do go out hiking, if I were you, I would not drink straight from a clear mountain stream. It's one of the reasons why hikers will carry water sterilization sets so they can basically get rid of this stuff so you can drink the water and not worry about getting this particular disease. You take a look at the scanning electron micrograph below, you realize that this organism is a very bizarre looking protist, and it's got several long flagella associated with its structure. There is a second one I need you to be aware of. It's called trypanosoma. Trypanosoma is a blood parasite. If you take a look at the picture at the bottom right, you will see what amounts to being these little purplish squiggly things in between red blood cells. That's a blood smear. And those squiggly things are the trypanosoma organism. Now, when they exist in a person's blood, they are competing for food molecules, they're competing for oxygen. As a consequence, the person who has this particular infection will be significantly fatigued to a debilitating extent. The disease is called African sleeping sickness, and the trypanosoma organism is carried by a biting fly called the tsetse fly, which you see pictured there at lower left. It is a significant problem on the African continent. Okay, the ciliates all belong in a phylum called the Ciliophora, sometimes called the Ciliata, and these single-celled organisms are characterized by short beating hairs, motility organelles, called cilia. And the classic example is paramecium, and you'll see that illustrated at the bottom left of the slide. As a matter of fact, there's a very, very nice light micrograph that shows you the teeny tiny little beating hairs around the periphery of the cell. Now, many of these ciliates exist as organisms living in the stomachs of a variety of animals. You see a cow there, and that's a fistulated cow. It's a cow that's got a hole in it. Where I was an undergraduate, we had a dairy husbandry major, and we had 1 or 2 fistulated cows. It's basically a hole cut in the side of the animal, and there's a portal that has a lid that you can unscrew, and you could actually reach your hand into the stomach of the cow, a particularly nasty experience, and extract samples from what's in that cow's stomach at the time. But if you take a look at the picture below the picture of the cow, you will see what amounts to being a bunch of teeny tiny little single-celled ciliated organisms that live in the cow's stomach. Cows are belonging to a group of mammals commonly called the ruminants. These are animals that have multiple stomachs. They chew the cud, so to speak, but inside of their stomachs, they have these little fermentation factories. And these little endocommensals, they live inside the cow. They don't hurt the cow. They benefit from living in the cow. The benefit does not extend to the cow necessarily, but they have a real nice place to live. Now, I should add this, and that is, as far as the protozoans go, ciliates probably contain the largest number of species. If I remember correctly, something like 8,000 currently described species of ciliates. Our last group of the protozoans are the sporozoans. And the sporozoans are the one group of protozoans that are not, are not classified on the basis of their motility structures. To review, the phylum Sarcodyna have pseudopodia. The phylum Mastigophora have flagella. The phylum Coleophora have cilia. The sporozoans are a unique group in the sense they are not classified on the basis of motility structures, but they are classified on the basis of their biology. You will see my first bullet tells me the sporozoans are sometimes also referred to as the phylum Apicomplexa. There's two alternative names, phylum Apicomplexa or phylum Sporozoa, regardless of the name that's used, and both names are in common use. These are intracellular parasites. In other words, they're really, really small. They're so small, in fact, that they can actually live inside individual cells. Case in point, we're going to take a look at the organism that causes malaria. I'm going to jump down to the very last bullet point so I can refer to the photograph. The photograph at the very bottom of the slide shows me some red blood cells. Those are human red blood cells. And you will see two of the red blood cells seem to be sick. They seem to be infected with something. As a matter of fact, one is partially open, and I'll see this little spherical thing inside of that red blood cell. That is an individual organism of Plasmodium vivax. Red blood cells are really, really small, which means that organism is really, really, really small. These are intracellular parasites. They're unique because generally they have multiple hosts, and as they go through their life cycle, they will demonstrate multiple stages. So, for example, the same organism I just mentioned, Plasmodium vivax, that causes malaria, is infecting human blood, but also infects mosquitoes. And so I see a mosquito. Now, it's not just any mosquito. It's a single genus of mosquito. As a matter of fact, it's not just a single genus of mosquito. It's only the females, because it's only the female mosquito that actually takes blood meals. So I've got a picture of a female mosquito here, and she's blood engorged. She is a member of a genus called Anopheles, and the Anopheles mosquito is the mosquito that is implicated in the transmission of the malaria organism. But so that female mosquito will take a blood meal, and she will, by virtue of biting the human, will introduce the malaria organism into the human. And if you take a look at the diagram immediately below the picture of the mosquito, you will see an inset illustration of the variety of life cycle stages that that organism goes through. And malaria is a really, really serious disease. It's not so big of a problem now, but the fact of the matter is historically, malaria has probably killed more people on the face of this planet than any other disease. All right, so let's wrap up here. The kingdom Protista contains the algae, which are classified on the basis of photosynthetic pigments. They contain the protozoans, which are largely classified on the basis of motility structures, and we covered the protozoans in pretty significant detail. We mentioned the fungal protists. Now, at this point, I just want to mention the fungal protists are organisms that actually resemble fungi in two respects. Fungi are filamentous organisms. We'll take a look at this when we get into our presentation on fungi. And fungi reproduce by making spores. These organisms are not fungi. They miss some specific characteristics that would make them fungi, but they superficially resemble fungi in terms of what they look like. They're filamentous. They reproduce by making spores, and they fall into two general groups of organisms, the water molds and the slime molds.